Hello, my name is A.Goeckner. I am a first-year PhD student in the University of Florida's Soil and Water Sciences Department. And today I'll be presenting on water column N2 saturation in natural and constructed subtropical ponds from down here in Florida. Urban stormwater ponds are constructed to provide services such as flood management and removing sediments and nutrients from stormwater runoff before that water is ultimately discharged downstream. They are one of the most commonly used stormwater control measures. This Sinclair et al. Study found that in the state of Florida, stormwater ponds cover up to 1% of the state's land area. And you can see that in the blue dots that cover the map. However, although they are widely distributed and are meant to remove nutrients from runoff, this Harper and Baker study from 2007 found that despite the water residence time from just a few to 600 days, stormwater ponds do not remove more than approximately 50% of nitrogen input through stormwater runoff. And so it goes without saying that we know little about stormwater pond biogeochemistry and drivers of nutrient removal or ecosystem functioning. N2 saturation in aquatic ecosystems can be used as a proxy for nitrogen removal or nitrogen pathways. So when we compare N2 saturation in water to the atmospheric equilibrium N2, we can tell if those waters are supersaturated or undersaturated in N2. So waters are supersaturated. That means that a lot of N2 producing activities are occurring, such as denitrification or anammox. If N2 undersaturation is being seen, then that is an indicator of N2 removal or N2 fixation. This recent study by Lokes, Johnson, and A.Goeckner looked at N2 saturation in lakes of the upper Midwest. What they found was that of 34 sites, a majority were supersaturated in N2, which means that anything with a value higher than 1 was supersaturated. And so this is showing that their lakes are leaking nitrogen, and that nitrogen fixation is not really compensating for nitrogen limitation. We were inspired by this study and wanted to apply it to urban ponds. And so we broadly asked, well, how saturated are stormwater ponds in dinitrogen? And does littoral vegetation make a difference? So urban stormwater ponds are typically constructed, such as the one on the left, with no vegetation implemented. And littoral vegetation is now being considered as a best management practice to look like the one on the right. But we know very little about the capacity of littoral vegetation to remove nitrogen or nutrients from stormwater ponds. Contrary to the Lokes-Johnson paper, we expect that saturation of N2 in urban stormwater ponds will be low, and that is because of N2 fixation by primary producers. We believe this because of a recent study that we conducted this last March in urban stormwater ponds. In this study, we did a nutrient addition bioassay where we treated water from a few ponds with nitrogen, phosphorus, and nitrogen and phosphorus treatments. After that incubation occurred and after nutrient uptake had occurred, we then analyzed that water for the rate of N2 fixation or the rate of denitrification. Shown here on the right are the results of that N2 uptake or N2 change. So here are the 3 ponds, and each one of these boxes is a different treatment. What we found was that 2 of these 3 sites were N and P co-limited, and this one was just phosphorus limited. In the treatments that received N and P, such as this light blue and this purple, or pink, those treatments that had the low NP had the highest rates of nitrogen fixation. So overall, everything below 0 is nitrogen fixation. So we know that the waters are capable of fixing nitrogen, and that low NP results in high N2 fixation activity. To conduct our study, we looked at 21 sites. 15 sites were urban constructed stormwater ponds from Lakewood Ranch, which is down in Bradenton, Florida at Point B. And we also wanted to compare urban sites to natural ponds and small lakes. And those 6 sites that we chose were from the Ordway-Swisher Biological Station, which is a large preserve. And from here on out, we will be referring to urban stormwater ponds as urban sites and these preserve sites as natural sites. We selected our urban stormwater ponds over a littoral vegetation gradient. So the percent of area that vegetation covered the pond, and that ranged from 0 to 35%. However, in this study, we have yet to see any correlation between vegetation and our data, and so we won't actually be talking about the vegetation part today. Finally, we conducted our sampling during a 2-week period of this last May 2021, last month, during an extended dry period that we had. And so we still expect to be seeing the nitrogen limited conditions that we saw in that March study I showed you in the previous slide. At each site, we characterized stratification, and then from 3 depths, we collected for surface, middle, and bottom waters. The figures on the top right show an example of where we would collect water from the water column. After water collection, we analyzed for N2 saturation via membrane inlet mass spectrometry, which estimates the concentration of N2 in water samples based off of its ratio to argon. Then we calculated an N2 saturation ratio by dividing our measured N2 by the expected atmospheric equilibrium N2. And so anything above 1 is supersaturated in N2, and anything below 1 is undersaturated in N2. Other things that we measured for were fluorescent DOM. So we looked at the degree of humic substances, and then other items that we will not be presenting on today are carbon greenhouse gases, nutrient ions, dissolved nutrient ions, and chlorophyll A. And today in this presentation, we'll be focusing on the N2 saturation and a little bit on the humic substances. So I want to bring up the Lokes-Johnson paper again. So this is their, this was the results of their study showing that a majority of their sites and samples were supersaturated. That was 85%. Well, in our sites, between natural and urban sites, 60% of our data was supersaturated in N2, and that's from all depths as well. However, when we split that data up between natural or urban sites, we saw that 100% of the data collected from natural sites, which includes all 3 depths, were supersaturated in N2. However, less than half of urban sites were supersaturated in N2. So they were primarily undersaturated in N2. This is what the data looks like when we split it up based off of water column depth. In natural sites, again, 100% of the data was supersaturated or above 1. Surface saturation was relatively similar to the saturation of the middle layer. And as one might expect, the bottom waters were always more supersaturated than their respective surface or middle layers, and so supersaturation increased with increasing depth. However, in urban sites, we saw a different pattern. Surface and middle waters were relatively similar to each other, but for bottom waters, out of 15 sites, only 5 sites had supersaturated bottom waters. And in many cases, the bottom waters would be undersaturated, and then their surface or middle waters of that site would be more supersaturated than the bottom was, which was really interesting. pH and dissolved oxygen can be considered indicators of primary productivity. So increasing pH, increasing DO, and thus increasing primary productivity was shown to have a negative effect on N2 saturation ratios. We believe that this is likely associated to increased N2 fixation with increased primary productivity. Temperature can also be associated with productivity. In our surface waters, temperature did not affect N2 saturation ratios, and that's likely because the surface waters are already very warm, so any increase in temperature likely doesn't have an effect. But temperature increases with importance with increasing depth, and they have a negative effect on the N2 saturation ratio. And I wanted to make this comparison to the Lokes-Johnston paper, where their temperatures were lower than the temperatures we saw, but they saw increasing or positive trends in N2 saturation with increasing temperature. So it looks as though in the upper Midwest, increasing temperature is having a positive or enhances denitrifying activities, whereas in our subtropical sites, increasing temperature, it may be enhancing primary productivity and N2 fixation activities. We also found some factors that influenced N2 saturation, but that were unique to whether it was a natural or urban site. In natural sites, factors that related to water clarity and stratification were positively correlated to the N2 saturation ratio. So here in this figure, I'm only showing a humification index, or an increasing degree in humic substances. But other factors that correlated in the same way were Secchi depth and the degree of stratification. In urban sites, the only other factor that we saw correlate with N2 saturation was conductivity. So here, increasing conductivity decreased N2 saturation ratios, and so we believe this is some sort of indicator of chemical stress. And we're hoping that after we analyze for our dissolved nutrient ions, we'll be able to tell a little bit more about what's going on here. But urban sites are subject to salt inputs. In summary, we believe that urban ponds are not indicating ecosystem scale denitrification. We believe this is because of high productivity that's going on and the potential nitrogen limitation that we're seeing during this drought period, which can enhance N2 fixation or can favor N2 fixing activity. We would like to, or we plan to sample these sites again after the wet season, and we expect that during that time we will see higher N2 saturations after landscape inputs of nitrogen from runoff. So, but we still have data to analyze from this last sampling, including the dissolved nutrients I talked about, dissolved organic carbon, carbon greenhouse gases that we collected from the same depths. We want to look more at the effect of stratification, and we'd like to turn those univariate relationships that I've shown here into a multivariate statistical analysis. Finally, I would like to thank the UF Center for Land Use Efficiency for funding this research, as well as the Ordway Swisher Biological Station for allowing us to do research on their site. And most importantly, I'd like to thank Annabelle and Katie, our interns, for helping me out with this project. Thank you very much.